Professor Sandvig is the next one. She comes from the academic hospital in Oslo. Very interested in the biology of EVs. Also will touch upon the issues in production of EVs, the effects of culture conditions on composition of EVs. So looking forward to your presentation.
Okay, thank you very much. Yeah, as you just heard, I'm going to talk about EVs, mechanism of formation, characterization, and possible use. Many of you may know that a cell is able to secrete different types of vesicles. There is pinching off of vesicles from the plasma membrane, and you have release of exosomes when the multivesicular body, which I can't point at, fuses with the plasma membrane and releases the exosome. And it's clear that there is both escort-dependent and escort-independent formation of vesicles in multivesicular bodies. The escort complexes are important in a variety of fusions and also in formation of the intraluminal vesicles. But I won't go into proteins today, but I will focus on lipids when it comes to EVs. And just to make sure, I show a sphingomyelin molecule. And as you just heard, neutral sphingomyelinase has been involved in creating interluminal vesicles in some cells, but it's only some cells apparently. And in that connection, it's important to note that sphingomyelin is normally localized to the membrane leaflet, which is not exposed to the cytosol, and it's actually not known to which extent endosomes have SM on the cytosol phase. It has to be very little. No one has really given an exact number. And recently, ceramide was found to be transported to the endosome by the protein SERT, and also exposed SM on the cytosolic side can be a danger signal and lead to LC3 modification. And then what happens is not really known. Different groups have very recently published that. So one has to do more in order to figure out the exact role of SM when it comes to formation of intraluminal vesicles in some cells. And if you want to see more about this type of discussion, you can find that in this paper, which you see below there, Lipids in Extracellular Vesicles, which was published now in June in Cold Spring Harbor Perspectives in Biology. And also when it comes to formation of exosomes or EVs from PC3 cells, there is extensive sorting of lipid classes. There is enrichment of 22 lipid classes in PC3 cells or exosomes, as you see in the figure. At the top, you see what's enriched in cells, and at the bottom, going to the right, you see what's enriched in exosomes. And there's also an extensive sorting of lipid species, not only lipid classes. There's a similar degree of up concentration from cells to exosomes of phosphatidylserine, 18,018,1, very long chain sphingolipids, and cholesterol. So it's tempting to speculate that these are sorted together into exosomes, making them tough and raft-like. So the data indicate that exosomes are raft-like, and this shows an illustration of a lipid bilayer of exosomes. And the illustration is based upon quantitative lipidomic data for exosomes released by PC3 cells. The quantification was done already in 2013. This model is much newer. And at the right, you can see the red sphingomyelin with long chains sticking into the inner leaflet where you have PS. And the amount of PS 18,018,1 could theoretically cover 80% of the area of the very long chain sphingolipids in the outer leaflet, assuming a diameter of 18 nanometer for the exosomes. So could the bilayer structure be stabilized by a handshaking between the very long chain sphingolipids in the outer leaflet and PS in the inner leaflet, perhaps even in a cholesterol-dependent manner? And in order to study that in more detail, we established a collaboration with Ilpo Vattulainen in Finland, and he looked at interdigitation between the two membrane leaflets. And here is one such model where you in the upper part have a sphingomyelin with a with long chain 24 zeros sticking into the inner leaflet. So this illustrates a handshaking that you might have when you have sphingomyelin with long chains. And he also demonstrated that the interaction between the two leaflets was increased when cholesterol is present. So that fits with the idea of the importance of these lipids to make tough exosomes. So it's clear that exosome EV composition, secretion, and uptake, it's not only cell type dependent, which it clearly is, but also factors like cell density can induce lipid changes in the cells. That we showed some years ago, and it was shown by MS, and one can expect that this will be reflected in the composition of the EVs and exosomes that are released. And also we have found that increased content of ether lipids in the cells leads to increased secretion of exosomes. And one can expect a change in their composition as well. So what affects release of exosomes from cells? I think it's important to get new assays. This shows one such assay. It's not perfect, but it might be useful. It's based on the use of radioactive cholesterol, but the hits have to be validated, clarified. If you look at the timeline down there, one can, for instance, incubate cells with siRNA, then add radioactive cholesterol, wash away that, and at some time point later, look at released radioactivity in order to get an idea about release of exosomes. And we used this method and found several hits, among them SNAP29 as snare molecule. And this was published now in June in Cell More Life Science, and the title is siRNA Screening Reveals SNAP29 Contributes to Exosome Release. And we did some studies of that, but I don't have time to go into that. Of course, when the EVs are released, they can be taken up by cells, as you just heard, and they are taken up by various endocytic mechanisms in the same way as nanoparticles can be taken in. And both type of vesicles might induce uptake by themselves, and this can be at least caused by binding and cross-linking of various molecules on the receiving cell. When we published this picture, now some years ago, we also published a toolbox of pharmacological inhibitors used to study endocytosis and some pitfalls that we see people often fall into. And I want to mention just one. That's methylbeta-cyclodextrin, which is used to extract cholesterol from cells. Of course, if you add it together with both cells and EVs, it will extract cholesterol from EVs as well. But if this compound reduces uptake of whatever you look at, it doesn't mean that caveoli are the ones which take up the compound. Caveoli are cholesterol dependent, but so are many other uptake mechanisms, including macropinocytosis. There is a revival of old ideas. People have suggested to use cholera B to label caveoli and their pathway. I don't think that's a good idea at all, because cholera toxin is also taken up by other types of mechanisms, different types of endocytosis, and also it causes signaling and changes intracellular transport. Then some questions related to cell biology and drug delivery. I think we need a lot of basic studies to know more about what we are doing. For instance, what happens when EVs run out of ATP, and when does it happen? Does the membrane then change? How soon does that happen? How can EVs best be stored? I want to mention that old knowledge has been forgotten. PBS may turn acidic during freezing and thawing, and that is certainly a risk when it comes to whatever you freeze down. It may change conformation, but if you expose it to pH like 5.5 or something. And also, one can ask, are the EVs produced or loaded with drug under conditions which activate scramblases in the membrane? For instance, calcium is reported to activate scramblases. Then you may expose PS, and you may get a more rapid removal of your EVs. And when adding drugs to EVs, how large a fraction of them turn inside out during addition of drugs? They are likely to behave differently if they turn inside out, partly due to exposure PS. And when adding drugs to EVs, how can one best determine and quantify drug just adsorbed at the surface versus what's inside? And will we be able to make reproducible batches? And how can we document batch reproducibility for products consisting of so many different molecules? I think you were touching on those questions as well. And then I've used my time, and I won't go through this. I already mentioned some collaborators. So thank you for your attention.